Intentional deep wall lift injections during endoscopic submucosal dissection. The senior author has carried out investigator-initiated studies with these companies in the last 2 years. However, this study was done independently without support or interaction with industry. Full-thickness perforations are a major complication that can occur when performing ESD or EMR. When they occur, surgery is often needed to repair or resect the large bowel. A tall mucosal lift is critical to the safe performance of ESD and EMR. The lift is created by injecting solution into the submucosal plane, which expands that layer and increases the distance between the polyp and the deeper bowel wall levels. This decreases the chances for perforation. Importantly, the polyp is detached in the widened submucosal plane. There are 3 principal ways to inject the bowel wall. The most commonly used method is a sclerotherapy needle, which requires a mucosal puncture. The second option is a punctureless high-pressure injection using either a machine or a hand pump. The third option is to use a combined needle knife and injection catheter, which allows the lift to be refreshed mid-case without an instrument exchange. For ESD and EMR, the goal of injection is to establish a submucosal lift which expands the layer the dissection will take place in, while also increasing the distance to the serosa, which reduces the chances of perforation. If the deeper layers of the bowel wall, namely the muscularis propria and the subserosal plane, are injected, they too can expand and will lift the mucosal polyp. Stable deep lifts can be made and can be confused with a superficial lift. Since deep injections don't expand the submucosal dissection plane, if ESD or EMR is attempted with a deep lift, it will be very hard to find and stay in the correct plane. Deep lifts are more likely to occur if a sclerotherapy needle is used. Deep lifts are less distinct, shorter, and wider. In contrast, a submucosal lift is usually round, tall, and has clear borders. To puncture the mucosa, a hard push is usually needed because the surface slides away from the needle tip. A powerful thrust commonly leaves the tip in the deep wall. When the injection is begun, the fluid will go into a deeper layer. The sclerotherapy catheter must be slowly withdrawn while injecting until a superficial lift begins to form, at which point the needle is held stationary and the injection continued. This human video clip shows a lift injection being made in the rectum. A diffuse low lift is initially obtained, but later a clear-cut superficial lift starts to form. This video of a sclerotherapy needle injection shows initial deep wall expansion. As the needle is withdrawn while injecting, a superficial lift begins to form. To further study bowel wall lifts, we made syringe injections into rectangular pieces of bovine colon. Here we see a superficial lift being made, after which the lift height is measured. Next, a scalpel is used to cut through the bowel wall, which allows the expanded layer to be identified. Here we see a superficial lift being made in a bovine colon. This is a cross-section through a superficial lift which shows the submucosal layer expanded. This is a histologic cross-section through a submucosal lift. The lift solution contained blue dye which can be seen in the submucosal plane. The muscularis propria can be seen below. Here is another section of a submucosal lift. The dye in this case was yellow and can be seen in the submucosal plane. This cross-section shows a mixed superficial and deep lift. The vertically oriented muscle fibers can be seen below the submucosal layer. This histologic section shows the blue lift dye particles in both the submucosal and the muscularis propria layers. Both the inner circular and the outer longitudinal muscle fibers can be seen. This lift cross-section shows expansion of the subserosal layer in addition to the muscularis propria and submucosal planes. The submucosal and muscularis propria expansion are more diffuse and less localized than the submucosal enlargement. This histologic section shows blue lift dye in all 3 planes. To get some idea of how much elevation is obtained from deep and superficial injections, a small study was done using the ex vivo bovine model. Bowel thickness measurements were made after intentionally deep and superficial injections. Cross-sections were then made to confirm which layers had expanded. This figure shows the contribution of deep and superficial wall expansion to mucosal lifts in 16 ex vivo bovine colon specimens. This small study verifies that stable deep wall lifts can be made and that the deep wall can notably expand. Deep wall expansion should minimize the risk of transmural perforation by further thickening the bowel wall. A widened muscularis propria could sustain a superficial partial thickness deep muscle injury without a perforation occurring. In the setting of ESD and EMR, intentional deep wall injections would make sense only in conjunction with a good submucosal lift, which is critical since that is the plane of dissection. Intentional deep wall injections make sense late during ESD cases because it is harder to refresh the submucosal lift when the polyp is mostly detached. Also, late in the case, submucosal lift duration is shorter than at the case's start. Intentionally injecting the deep wall as well as refreshing the submucosal lift is therefore logical in an effort to avoid perforation. Here, using the bovine model, we see a polyp late during ESD when it is largely detached. The deep wall is being injected prior to the final excision. The exposed deep muscular surface can be seen to expand in response to the injection. If during the final detachment the muscle is injured, perforation is less likely. Here we see an additional video clip showing an injection being made directly into the muscularis propria in bovine colon after an ESD has been performed. The expansion of the deep layers can clearly be seen. To summarize, the muscularis propria and subserosal planes can expand when injected. When sclerotherapy needles are used, after insertion, the needle tip often resides in the deep wall. The initial injection may expand the deep layers, and a stable deep wall lift may develop and be confused for a submucosal lift. Prior to ESD, the lift must be critically assessed to be sure the lift is submucosal. If not, a more superficial injection is needed. Intentional deep wall injection and expansion in conjunction with the submucosal lift should decrease the risk of perforation during ESD or piecemeal EMR. This makes sense especially later in the case. The authors are now using this approach clinically.